Hello, everybody, and welcome to MCAT Metabolism Part 5. We'll be finishing up our metabolism lecture series today. So here's a look at what we've covered already. We covered the digestive enzymes, carbohydrate metabolism. We covered the pentose phosphate. We had an electron transport chain. We covered lipid metabolism. There's one more piece of lipid metabolism we're going to talk about today, which is steroid metabolism. We'll talk about very briefly. And then ketone bodies are going to be related to lipid metabolism. We'll cover the glucogenic and ketogenic amino acids, and the urea cycle will be sort of in very, very broad overview detail. So I'm not going to spend too much time on this slide. If you're looking at part 5 of our lecture series, you've probably heard me go through this 4 times already, so I won't belabor any points here. All right, so steroid metabolism, our first out of our 4 topics for today. So steroid catabolism, breaking down steroids in animals, is incomplete, owing in part to steroids' hydrophobicity. So because they're very hydrophobic, it's harder to get these guys accessible to our enzymes, and also particularly because they're so nonpolar, lots of carbon and hydrogens, it's hard to functionalize steroids. Steroid catabolism will begin with cytochrome P450 enzymes in the liver. So you do want to be familiar with the cytochrome P450 enzymes because the MCAT does love to ask about them. Catabolism will be upregulated by high levels in the bloodstream of steroids. And just recall, so cytochromes are heme-containing enzymes that perform redox reactions. Of course, cytochrome C in our electron transport chain. Cytochrome P450s are monooxygenases. So they are enzymes that oxidize their substrate. And then as these steroids begin to be metabolized, they're processed into bile salts, which, as we know, will emulsify dietary fat droplets in the small intestinal lumen. That'll be one purpose of bile salts. The other purpose of bile salts would be to eliminate cholesterol and bilirubin from the gut. So bilirubin, if you haven't heard of that before, is partially catabolized heme from aged red blood cells. So as red blood cells get older, they don't have a nucleus, so they're not capable of transcription or translation. And they will get older over time, and then we will recycle a lot of the components. And one of the components that we start to metabolize is heme from the hemoglobin. And when it begins to be metabolized, becomes bilirubin, which is the chemical that is responsible for jaundice. So in people with liver failure, they can't produce bile, and therefore they can't eliminate bilirubin from the body, because this is the process by which bilirubin is eliminated from the body. So the yellow color of a jaundiced patient is due to buildup of bilirubin, and typically we see that with people who have some type of liver failure, which could include alcoholism. It's a little more on steroid catabolism. Cholesterol, when it begins to be metabolized, will become cholic acid, which is a bile acid. Bile salts are made up of an acid and a metal ion, such as sodium. We can see sort of what was once the side chain sort of cholesterol. And steroid hormones, on the other hand, such as like testosterone, aren't going to have this side chain of cholesterol. So we're not going to be able to oxidize them in the same way to a carboxylic acid. Steroid hormones, on the other hand, when they are ready to leave the body, will be hydroxylated or sulfonated. So that would increase, or combined with glucuronic acid. And what that would do is that would serve to increase hydrophilicity, increasing water solubility, which will allow them to be excreted from the urine. Any questions on this slide? All right, and then steroid anabolism. So there's a few things that we want to take away from this slide. Steroid anabolism begins with 3 molecules of acetate, which will condense into mevalonate. There's a passage on mevalonate and some of these steroid intermediate molecules in, I want to say, one of the AMC full lengths, I believe, in the sample. There might be full length 1 in the ChemPhys section. So this isn't something that you should be able to know in great detail, just sort of recognizing some of these names, such as mevalonate, such as acetate. Acetate we see in a lot of different places. Mevalonate becomes decarboxylated, so we'll lose this carboxylic acid as carbon dioxide. And then it will also be dehydrated. So we'll lose a alcohol and a hydrogen from the carbon over here, giving us an isoprene. And we recognize, or we know isoprenes are from terpene metabolism. So isoprene is the simplest terpene unit. And we call this activated isoprene because in the process of synthesizing this, it's called isopentyl pyrophosphate. We're adding two phosphate molecules or a diphosphate onto the isoprene. So then when it arrives at the next enzyme, it's already carrying some free energy. Remember that we know hydrolysis of ATP or other nucleotide phosphates leads to a release of energy that can be used to couple reactions. So for instance, in this case, instead of having an ATP that we're coupling the next step of the reaction with, we're already giving the phosphates to the substrate. So the substrate's like BYOB, the substrate's bringing its own energy to the party. So multiple, and this is also known as isopentyl pyrophosphate. Remember, pyrophosphate refers to a diphosphate, so two phosphates that are linked together. Then isoprenes will be polymerized into squalene, and the squalene will be cyclized to form cholesterol. So you will see squalene pop up on some of the AMC questions. Now, they're not expecting you to have memorized this pathway for sure. However, it is good to be able to recognize these because you get to your point, you get to your correct answer choice faster. Any questions on steroid anabolism? So then cholesterol can be functionalized into a variety of different steroids. All right, so a little more on isopentyl pyrophosphate. And this slide is not for you to memorize. Isopentyl pyrophosphate is the intermediate in cholesterol anabolism. So here's our isopentyl pyrophosphate. We called the deactivated isoprene before. And then cholesterol, which can be made into steroid hormones, as we said, bile salts, and as well vitamin D. There are a lot of other places that isopentyl pyrophosphate can go. So this is just for your own curiosity, satisfying your own curiosity here. You can see vitamins A, E, and K, carotenoids, such as like carotene, which is what gives carrots their orange color, can be made into rubber, interestingly. It can be made into some familiar quinone electric carriers, such as ubiquinone, right? So this is also where we get our ubiquinone from. And again, this slide is not for you to memorize. This slide is just for a little bit of extra background. Questions? Okay, so that's it for steroids. On to ketosis and ketone bodies. So the brain and the central nervous system run primarily on glucose catabolism. When blood glucose is low and glycogen is depleted, which is typically 24 hours into starvation, ketone bodies get produced in the liver and sent to the brain. And so you may have wondered, as I wondered, why does the brain and the central nervous system run primarily on glucose? And the old thinking, and this should be fairly logical, like we're going to give them some credit because this is a fairly logical way to think about it. Usually if something doesn't go into the brain, right? Because it can't cross the blood-brain barrier. The blood-brain barrier is impermeable to a lot of different molecules. And so this was the old way of thinking, was that they were just too hydrophobic, these fatty acids, to cross the blood-brain barrier, or something along those lines. The new thinking is a little bit different, a little bit more nuanced, which is that in the brain, the beta oxidation enzyme equipment is down-regulated by evolution. There's a couple of plausible reasons for this. I'm not sure if we would call this the new hypothesis or the new theory. I don't know if it has enough research support to be called a theory yet. So the new thinking is that, so the reason that the beta oxidation enzyme equipment gets downregulated in the brain is that oxygen consumption during beta oxidation. So remember, beta oxidation, we need lots and lots and lots of oxygen because we're getting all of our energy pretty much from the electron transport chain. We're making NADH and FADH2 every time we perform a round of beta oxidation. And at the end, we're going to have acetyl-CoAs, which are going to enter then the citric acid cycle. We're going to get more NADH, FADH2, as well as some GTP. So to get energy to get ATP out of all of these reduced electron carriers, we have to send them through the electron transport chain. And because fatty acid oxidation, everybody's more reduced, so you have to do even more oxidation than you do with carbohydrates. The level of oxygen consumption during beta oxidation risks the neurons becoming hypoxic. And it should be pretty self-evident as to why that would be bad. We don't want our neurons to die. We need those guys. Those guys will not divide again, at least canonically, they won't divide again. And then superoxide production is another reason. So we didn't really go into this whole idea of reactive oxygen species being produced during beta oxidation. But long story short, there are some reactive oxygen species produced during beta oxidation. And this risks oxidative stress and apoptosis of neurons, which again, that would be very bad. We want to preserve as many neurons as we can. These are the reasons why it's thought now that the beta oxidation enzyme equipment was evolutionarily downregulated in the brain. There's 3 ketone bodies. You do need to know the names and the structures of all 3 ketone bodies for the MCAT. There's acetone, acetoacetate, and beta hydroxybutyrate. And we'll look at those structures on the next slide. Any questions before we head deeper into ketosis? Moving on. So here are structures for our ketone bodies. We have acetone, which you're probably familiar with, being the simplest ketone. Does anybody remember or know where we encounter acetone in like everyday life, particularly if you're somebody, in the labs for sure. Yeah, that's our everyday life. And acetone has like a sweet, fruity smell. It's present in nail polish remover. So if you smell nail polish remover, that strong, sweet, and somewhat fruity smell is due to acetone. It's a great solvent because it can help solubilize organic molecules. And the issue with acetone is that because it has very weak intermolecular forces compared to our other ketone bodies, is that as soon as it's produced, it's just going to be exhaled on the breath. And so we're not going to actually ever be able to get any energy from acetone. It's kind of the useless ketone body. Then we have acetoacetate. You could also call this beta ketobutyric acid. So we have a 4-carbon molecule, which is a carboxylic acid on the terminus, and then has a beta keto group. So that's our acetoacetate. And it is formed by condensation of 2 acetyl-CoAs, as we'll look at on the next slide. And then our third member of the party is beta hydroxybutyrate. You also may see this said as 3-hydroxybutyrate, which of course would be because carboxylic acid gets the 1, 2, and 3, or via our Greek nomenclature, we could call beta hydroxybutyrate. Beta hydroxybutyrate is the predominant ketone body in the bloodstream. It's less reactive than acetoacetate, and so it's a good place to send our energy through the bloodstream since it's less reactive and get it to the brain. Any questions on introducing our 3 ketone body friends here? All right. So ketone body formation, we will be forming ketone bodies starting from 2 acetyl-CoAs. And in terms of locations, so where are we right now? We are in the liver mitochondria. So ketone bodies are produced in the liver. And does anybody recognize this thiolase from this first step of ketone body formation? Where did we encounter that before? Which metabolic pathway involved thiolase? And notice that there are 2 acetyl-CoAs here. Thiolase catalyzes actually the last step of beta oxidation, the final step of a round of beta oxidation. In the final step of beta oxidation, right, we have thiolase is going to lead to breaking off of 1 acetyl-CoA. And in the very last round of beta oxidation, where we're going from a 4-carbon fatty acid to 2-acetyl-CoAs, that very last step of the last round of beta oxidation is actually the reverse of this step right here. So when we're making ketone bodies, our first step is the condensation of 2-acetyl-CoAs to form acetoacetyl-CoA, which is performed by thiolase, as we said, is the reverse of the last step of beta oxidation. Now, the next few steps here are not super important with this HMG-CoA synthase and HMG-CoA lyase. What's going to happen is they're going to add another acetyl-CoA to the molecule in blue here, which is going to lose its coenzyme A. And then we're also then in the next step, the lyase is going to eliminate a acetyl-CoA. So the net reaction is actually simply a water coming in and a coenzyme A coming out. So if we look over here, now it's not the exact same carbons, but for our purposes it is. Our carboxylic acid, or sorry, a thioester became a carboxylic acid over here. So we go from acetoacetyl-CoA to acetoacetate. In other words, the net reaction here is going to be hydrolysis of coenzyme A from acetoacetyl-CoA to form acetoacetate, which is our first ketone body. Once we have acetoacetate, there's two pathways we can go. Oh, and then synthases and lyases. What class of enzyme are those? Well, of course, the lyase is a lyase. And synthases are also lyases, so both enzymes are both lyases. This is kind of similar, or I kind of draw a connection here between this step, or these couple steps here with step 2 of the citric acid cycle, which is a aconitase. If you recall back to our citric acid cycle lecture, a aconitase is a lyase that first eliminates a water from citrate, and then hydrates, instead of leaving a double bond, and then hydrates the double bond on the next carbon down to form isocitrate. So aconitase is a lyase that catalyzes a net isomerization reaction. So this pair of lyases catalyzes a net hydrolysis. Okay, and then as mentioned, once we get to acetoacetate, we have two pathways. We can do a spontaneous decarboxylation to acetone, which, as we said, is just quickly exhaled on the breath. We never get any energy from acetone, so it's kind of a useless ketone body. There's also an enzyme here, acetoacetate decarboxylase, which would catalyze this reaction. Now, not really sure what the main purpose of this enzyme would be. Maybe to prevent having too much ketone bodies, and then some of the ketone bodies will leave off the breath as acetone, but that would just be a guess for me. The other pathway acetoacetate can go is it can be reduced to beta hydroxybutyrate, which, as we said, is going to be the major ketone body in the bloodstream. And then, as we can see, we have a carbonyl, and we use NADH to reduce the carbonyl to an alcohol. So one of our themes of metabolism was when you oxidize a carbon to oxygen or carbon to sulfur, you get an NADH produced via reduction. And so this is kind of the opposite. This is similar to lactate dehydrogenase, where we use an NADH molecule to reduce a ketone to an alcohol. Any questions on this slide of ketone body formation? All right, so the important takeaways here, I would say, are acetyl-CoAs, which can be produced by a beta oxidation, are condensing via thiolase into acetoacetyl-CoA. The next two steps are not really important, but you should know that acetoacetyl-CoA will become acetoacetate, and then can either go this way, be decarboxylated acetone, or can go this way, be reduced into beta hydroxybutyrate. So those would be our important takeaways from this slide here. All right, so we went over how our ketone bodies are formed. Now let's talk about how does the brain actually use these guys for energy. So now we're in the brain mitochondrial matrix, or in the neurons mitochondrial matrix. And beta hydroxybutyrate, once in the brain, gets oxidized to acetoacetate. So now we're going to go the opposite way that we had gone previously in ketone body formation. We will oxidize the carbon to oxygen bond, and we will reduce NAD plus to NADH. So you're going to get a very a relatively small contribution of energy just from this NADH as that goes into the electron transport chain. Once we have formed acetoacetate, we'll actually reform acetoacetyl-CoA. Remember, that was our intermediate before in the formation process. And this is done by a coenzyme A transferase. Interestingly, the donor here is succinyl-CoA becoming succinate. Normally, what is the enzyme that catalyzes this reaction in the citric acid cycle? Succinyl-CoA to succinate. So normally that would be succinyl-CoA synthetase. And do we get any energetic byproducts of this reaction when it's performed in the citric acid cycle? Do we get an NADH, FADH2, ATP, GTP? Yeah, normally we would be getting a GTP from that step. But if we instead send the coenzyme A to acetoacetate to make acetoacetyl-CoA, we miss out on the GTP. But that's not going to be a huge problem because we're going to get a lot of energy down the line here. We're converting now to a thioester with coenzyme A. And acetoacetyl-CoA will then undergo that opposite step of ketone body formation, the first step in the opposite direction, where we'll have thiolase, a transferase, transfer a coenzyme A to the beta carbon, the ketone here, leading to two acetyl coenzyme A's. And this is where the brain's going to get the majority of energy from the ketone bodies from, because once we have those acetyl-CoAs, they can enter the citric acid cycle, and each one of them would give us 3 NADH, 1 FADH2, as well as 1 GTP. So that's how the brain utilizes these guys for energy. Any questions on this slide? Moving right along. So I find this a good place to talk about diabetic ketoacidosis, also known as DKA. This is not a pathophysiological mechanism to know in great detail. This is mostly just to reinforce the various parts of metabolism that are involved here so that we can kind of get a clinical scenario for how our different parts of metabolism interact in ketoacidosis. Maybe to remember, so we have two types of diabetes mellitus, two types. We have type 1 and type 2. Does anybody remember the difference between type 1 and type 2 or how either of those work? Type 1 has no insulin, good. So we call type 1 insulin deficient and is genetic, right? So type 1 diabetics have a genetic predisposition towards autoimmunity, typically towards their beta cells. So they are not able to make any insulin. Whereas in type 2 is insulin resistant, good. In diabetes type 2, we are able to produce insulin in our beta cells of our pancreas. But the insulin receptors, due to we're talking years and years of high levels of insulin, typically related to lifestyle, diet, years and years of high exposure to insulin lead to the receptors being less sensitive to insulin, and we call that insulin resistance. So in other words, we could say that in diabetes type 2, insulin levels are present, but they're insufficient for glucose uptake because the cells have become resistant. Now, so this is going to be important because with insulin is usually present post-meal, right? After we've had a meal, our pancreas will produce insulin to tell our cells, hey, there's an abundance of energy. We need to store some of this energy so that we can use it when we don't have an abundance of energy. But I like to think about this as sort of a communication breakdown. So the bloodstream's saying, hey, got lots of glucose, got lots of biomolecules from our food. Can we go ahead and store some of these possibly? And the cells are like, new number who this. And so therefore the cells actually think that the bloodstream is hypoglycemic. So because they're not receiving any glucose and they're not responding to insulin, the cells think that the bloodstream is hypoglycemic, particularly in the liver. And so they're going to do gluconeogenesis, because also gluconeogenesis is inhibited by insulin. So what we could say is happening here is that gluconeogenesis is unnecessary. We don't need to make glucose. The problem is getting glucose into the cells, as well as overactive. So how does this interact with our other pathways? If we're doing too much gluconeogenesis, remember that one of the gluconeogenic precursors is oxaloacetate. So if we're not doing a lot of glycolysis, because we just simply can't do glycolysis without glucose, and we're doing gluconeogenesis, then our source of precursor molecules cannot be pyruvate. It has to be citric acid cycle intermediates. So in these liver cells, when they're doing lots and lots of gluconeogenesis, which is unnecessary and overactive, they're depleting their citric acid cycle pool of intermediates. So simultaneously, remember that when there's lots of glucose uptake, right, into the cells, and we're doing lots and lots of glycolysis, we'll have a buildup of pyruvate, and we'll have a buildup of acetyl-CoA, and the cell will be like, hey, I've got an abundant amount of energy. I should start making some fatty acids. And so acetyl-CoA will become malonyl-CoA, and particularly, and remember, malonyl-CoA is our precursor for fatty acid synthesis. One thing we said about malonyl-CoA is that it inhibits the carnitine shuttle, so that therefore, if we're trying to do fatty acid synthesis, which would be why we have malonyl-CoA present, we wouldn't simultaneously be doing beta oxidation. We wouldn't have both processes running at once. But because we're not having enough glucose uptake, the cell is not triggered or instructed to make fatty acids happen, there's low levels of malonyl-CoA, and beta oxidation of fats increases. And this is helpful for the cell because the cell isn't getting a lot of glucose, so it's going to start to do beta oxidation. But the problem is the acetyl-CoA that we get from this beta oxidation doesn't have receiving molecules. It doesn't have enough oxaloacetate to combine with and enter the citric acid cycle, because remember, we've depleted our citric acid cycle with all this gluconeogenesis that we're doing. So then, because the citric acid cycle pool can't accept the resulting acetyl-CoA, we will form ketone bodies. So this will trigger ketone body synthesis. And so we don't necessarily need those ketone bodies. Right? The problem is not that we don't have enough glucose to feed the brain. The problem is that there's a communication breakdown where the brain and the neurons are not able to take up the glucose. So we have not only hyperglycemia, but we also have ketone body synthesis, ketosis. And when these guys get into the bloodstream, there's going to be a buildup of especially beta hydroxybutyrate. And this is what leads to diabetic ketoacidosis, is this excess amount of carboxylic acid in the bloodstream due to unnecessary ketone body synthesis. That's the pathophysiology of DKA. Any questions here? Very interesting, very exciting to see so many pathways linking up in this metabolic disease, right? We had fatty acid synthesis, beta oxidation, glycolysis, gluconeogenesis, citric acid cycle, and ketone bodies are all sort of interacting here. And so this could happen in type 2 diabetes and type 1 diabetes. I actually think it happens more in type 1 diabetes. And remember, the problem with type 2 diabetes is there's a communication breakdown due to the receptor's insulin insensitivity. Whereas in type 1 diabetes, we're simply not getting any insulin. So we're gonna have the same problem happen, and probably to even a greater degree, because not only can the problem is not that they can't respond to any insulin, or they have less responsiveness to insulin, the problem is there isn't any, right? All right, so then that brings us to our third topic of the day, which is glucogenic and ketogenic amino acids. So amino acids can be converted to a wide variety of biomolecules. One of our sources of amino acids is dietary. So we get abundant amino acids in the diet, especially the American diet is very proteinaceous typically. has a lot of everything. We would have during starvation. So this is after we've depleted all of our liver glycogen. This is after we've depleted our gluconeogenic precursors. So gluconeogenesis is stalled. Glycogenolysis is pretty much depleted in the very first day. So like 5 or so days into starvation, the body's own muscle starts to break down. That's why if you see somebody who's in starvation, they're not only very skinny because they're depleting their fatty acids. Fatty acids would come before muscles. But they've already depleted all their fatty acids. They're very skinny, but they're also emaciated. They don't have muscles where they should have muscles because the body has started to break down those muscles for energy into amino acids. And another thing we can do with amino acids is we can generate non-essential amino acids from essential amino acids. So does anybody remember what tells us or what's the difference between essential and non-essential amino acids? Yeah, essential amino acids have to be consumed in the diet. They cannot be produced via de novo synthesis. We can't synthesize them from scratch. Good. And then non-essential amino acids are non-essential because of this fact, because they can be made from the essential ones that we obtain from the diet, or they can be synthesized elsewise. The first reaction of catabolism of amino acids is transamination, in which the alpha amino group, so the N-terminus, is transferred to an alpha ketoglutarate to should say 2 alpha ketoglutarate to form glutamate. And we'll look at this reaction on the next slide. 2 amino acids are exclusively ketogenic. So when we say an amino acid is ketogenic, that means it can be made into acetyl-CoA. 13 amino acids are exclusively glucogenic, so they can only become glucose. And the remaining 5 amino acids can go both ways. They can be glucogenic or they can be ketogenic. That's our overview here. Any questions before we head into it? All right, so one of the things you'll have to do for the MCAT is memorizing the categories of ketogenic and glucogenic amino acids. So there are some mnemonics out there. I have my own mnemonic, which I'm going to share with you. And so it's probably easiest to memorize the 2 and then memorize the 5 and then classify the rest as glucogenic. So that's how we're going to go about this. So with ketogenic amino acids, we have the ketogenic lovers, K and L, lysine and leucine. And so I love this mnemonic because it's literally KL, ketogenic lovers are lysine and leucine. So these guys really love each other. They have very similar sounding names. Do you know that you're more likely to end up marrying or being with somebody who has a similar name to you? And then the 5, the glucogenic and ketogenic, the ones that can go both ways. We have the aromatic love triangle of our tryptophan, phenylalanine, and tyrosine. And then because there's 3 of them, we have threonine, 3 aromatics. And then the last one is isoleucine. And isoleucine is isolated from leucine. It is the isolated lover. It doesn't fit with anybody here. It misses its buddy leucine over here. So that's how, my mnemonic goes for the ketogenic, glucogenic amino acids, and the rest will be glucogenic. So then you don't have to memorize the 13. Any questions here? And so then here's our diagram of the glucogenic and ketogenic amino acids. So before anybody freaks out on me, we're not going to memorize, and you're not to memorize all these connections. Just memorizing the categories and knowing what the categories means is sufficient. So what makes a ketogenic amino acid ketogenic? We said that they can become acetyl-CoA. So lysine and leucine actually can only become acetyl-CoA. And then they can combine with the citric acid cycle pool to form citrate, isocitrate, alpha ketoglutarate. And by the time we're down to succinyl-CoA, we'll have lost 2 CO2s. And so we will have lost both of the carbons that were acetyl-CoA. Then our glucogenic amino acids. So glucogenic amino acids can only become either pyruvate or a citric acid cycle intermediate. And again, you don't have to memorize these connections, just know the categories and what the connections mean. The reason why these are glucogenic is because when they become a citric acid cycle intermediate, my camera's glitching out on me. 1 second. When these guys become a citric acid cycle intermediate or a pyruvate, but the citric acid cycle intermediates can then become oxaloacetate, which can become PEP, which can then become glucose. That's why these are glucogenic, is because we can have them enter into gluconeogenesis at no cost of citric acid cycle intermediates. So we can have them just add to the citric acid cycle pool by becoming an intermediate. So we're adding on a citric acid cycle pool intermediate. Then we could just use that citric acid cycle pool intermediate to then become phospholipopyruvate through oxaloacetate and then up the chain of gluconeogenesis all the way to glucose. So why do we not consider lysine and leucine to be glucogenic amino acids? Because they can become acetyl-CoA, right? And then from acetyl-CoA, when it enters the citric acid cycle, can then become, one can eventually produce oxaloacetate, which can then become PAP and glucose. So then why do we call these not glucogenic? This is a tricky one, but follow this. If we were to add lysine, leucine, or any of these amino acids, have them become acetyl-CoA and add that acetyl-CoA to oxaloacetate, we won't have actually added an intermediate of the citric acid cycle. We'll have used an intermediate, oxaloacetate, to form citrate. So we're not actually adding to the pool, if that makes sense. And then if we go over here, we'll lose both carbons of acetyl-CoA. And then if we wanted, so we've now gained 2 carbons of the citric acid cycle and lost 2 carbons. So then if we wanted to make glucose, we would use an oxaloacetate. But then we didn't actually gain anything in our citric acid cycle pool. So if we tried to make glucose from these ketogenic amino acids, we would lose from the pool. And that wouldn't be good because then we have less citric acid cycle intermediates to accept more acetyl-CoAs. So that's why we would not consider lysine and leucine to be also glucogenic. Does that make sense? Any questions here? Sweet. So our first reaction here is transamination. So as we said before, the first step in amino acid catabolism is transamination. And when we have, so here's our amino acid will become deaminated. We'll lose the amino group, which will be transferred to alpha ketoglutarate, turning alpha ketoglutarate into glutamate, and then turning the amino acid into an alpha keto acid, which then can, depending on the amino acid, will undergo a series of different reactions, eventually becoming either acetyl-CoA or a citric acid cycle intermediate or pyruvate. So the name here is just generic. It's the aminotransferase. So aminotransferase can work on a lot of different amino acids. And the prosthetic group here, PLP, is called pyridoxal phosphate. So pyridoxal phosphate is it's a good prosthetic group to know or to be familiar with because there is actually a passage about this process of transamination. Now the focus is on organic chemistry, so you don't have to come into this passage. It's in the official guide, Chem Phys, passage number 1. So you don't have to come in with knowledge of transamination or glucogenic and ketogenic amino acids to tackle that passage, because again, the focus is OChem. But if you were to get a passage like that, it can only serve you, it can only help you to have that additional context. So that's pyridoxal phosphate, and its job is to just temporarily hold on to the amino group from our alpha amino acid, temporarily holds onto it, and then give it over to alpha ketoglutarate. Here's our structure of pyridoxal phosphate. I believe it's one of the vitamin Bs. It's like vitamin B6 or something like that, or B5. And so during this transfer, as you'll see when you do that official guide passage, during this transfer, the amino group will form, that's why this is highlighted, will form an imine with this aldehyde over here. And then the imine will revert back to a carbonyl when it gives the amino group to alpha ketoglutarate, becoming glutamate. So this is transamination, and this is the only step of amino acid catabolism that you really should know. Questions here? All right. So this is just I saw this when I was reading in the Leninger textbook about aminotransferase. And so this is a convenient, good place to talk about enzyme mechanisms. So aminotransferases are a classic example of ping-pong enzyme metabolism. So what do we mean by that? In the ping-pong enzyme mechanism, what happens is the first substrate, which is in this case the amino acid, enters the active site. The first substrate has to react with the enzyme, modifying both substrate and enzyme, and must leave before the second substrate, alpha ketoglutarate, can enter. So that's the ping, first thing comes in, gets modified, and then the pong, second thing comes in, gets modified, and the enzyme goes back to its resting state. So in other words, ping-pong enzyme mechanisms don't have a ternary complex. What do we mean by a ternary complex? So ternary complexes are when we have an enzyme with two substrates. So typically, for like example, like a transferase, you might have a transferase which has the two substrates both present in the active site, transfers something from one substrate to the other substrate, and then allows them to both leave. So that'd be a ternary complex, as opposed to what we're describing here, where one thing has to come in, the other thing has to leave. The first thing has to leave before the second one can come in. So I do have a practice problem, and the reason why I bring this up in a seemingly unconnected place is just a great place to talk about ping pong versus what we call sequential enzyme mechanisms, and this does show up on one of the AMC fault lengths. Let me erase the answers before I show you. Okay. So here is going to be the setup for our problem here. So we have here our Western blots, and we have some hypothetical enzyme that is a 60 kilodalton enzyme. And we have two substrates. One is 10 kilodaltons, and the other is going to be 5 kilodaltons. So then we have 3 different gels for 3 different unknown enzymes. And let's start with our first group right here. So we have 5 bands here. We have a 5 kilodalton band. We have a 10 kilodalton band. And of course, those are going to correspond to our substrates. We have substrate 1, which is 10 kilodaltons. And we have substrate 2, which is 5 kilodaltons. We have a band at 60, representing our enzyme. So question for you. What would the 65 kilodalton band represent? So the game here is that we have this enzyme, we have 2 substrates, and we have some inhibitor, which is of comparatively negligible mass. We have on the order of daltons versus kilodaltons here. And so this inhibitor assume that this inhibitor is able to just grab onto the enzyme, whether it's free enzyme or whether it's enzyme bound to substrate, and lock everybody in place such that we can get discrete bands based on who is bound at any given time during the reaction. So who would all be present in the 65 kilodalton band? The enzyme and substrate 1, enzyme substrate 2, enzyme substrate 1 and 2. Enzyme and substrate 2, good. So we have enzyme and substrate 2. Substrate 2 is 5 kilodaltons. What about our 70 kDa band? What would be present there? Enzyme and substrate 1. So in this gel, did we have a ternary complex form? We have a ternary complex form. Because what would be the molecular weight of a ternary complex? Be 75. So then if no ternary complex formed, then would this be ping pong? Based on our definition of ping pong, if there's no ternary complex forming, it must be ping pong, meaning that one of these substrates will have to enter the enzyme's active site, be modified, and then leave before the other substrate comes in. And we don't know particularly which one comes in first or second, just based on this information here. What we do know is that they're never bound at the same time. This would be describing a ping pong enzyme mechanism. So that's our first type of enzyme mechanism. Our second category is going to be sequential. So in sequential enzyme mechanisms, both substrates must be present simultaneously in the active site. So there is therefore a ternary complex in sequential enzyme mechanisms, where we'd have E, substrate 1, and substrate 2. There's two types of sequential enzyme mechanisms. There's ordered, where one substrate must enter the active site first. So ordered sequential, the one has to enter, and then in contrast to ping pong, it stays there for the other substrate to enter. And then there's also random order sequential, which is that the order S1 and S2 enter the active site does not matter. So S1 could come in first sometimes, S2 could come in first sometimes, and it wouldn't matter. Okay. So keeping those in mind, we look over at our second gel. So we have, of course, our substrate 2, our substrate 1. We have our enzyme at 60. We said 65 represents enzyme and substrate 2. The 70 represents enzyme and substrate 1. So 75 would represent what? '75 would have to represent our E, S1, and S2, our ternary complex. Okay, so because there's a ternary complex, this must be a sequential mechanism. Now, would this be ordered sequential or random order sequential? Would this be ordered sequential or random order sequential? How do we know? Based on our data, did one substrate have to enter first, or did it not matter which one entered first? It doesn't matter, right? We saw both. We saw a 65, which was E substrate 2. We saw a 70, which is E substrate 1. And we found a 75, which was E substrate 1 and substrate 2. So it turns out for this one, it must not matter the order in which substrates bind. So we could call this random order sequential. Right, and then let me move these bands, so it's 60, 70, 75, so they match up a little better. Go to 60, 70, and 75. And we can identify all of our bands, substrate 2, substrate 1, enzyme. With 70 was enzyme and substrate 1. And our 75 was the ternary complex. So for a third enzyme, what would be the mechanism? The ping-pong, random order, sequential, or ordered? Yeah, this would be ordered sequential, because who has to bind first, apparently? Yeah, apparently S1 has to enter before binding to S2. So those are the three types of enzyme mechanisms. Any questions here? Anything I can clarify? Yeah, so when you see that, enzyme mechanism question, you're going to get it right now. Yeah, I'll give you a chance to take a screenshot if you'd like to. 5, 4, 3, 2, 1. Transaminase. Oh, actually, we're done with glucogenic and ketogenic amino acids now. So any questions before we head over to our last order of the day, which is urea cycle? All right. So the urea cycle, now for the urea cycle, you really just want to know the overall purpose of the urea cycle, the location, and a couple of involved molecules. The urea cycle, so the accumulated amino groups from transamination of amino acids must go somewhere. Ammonia, NH3, is extremely toxic to animals, so release as ammonia is not an option. And some ammonium is also acquired from bacterial oxidation of amino acids. And likewise, we have to figure out something to do with that, because we can't have free ammonia in the body, it's very bad for us. So that's what the urea cycle comes in. The urea cycle occurs in the liver. And the net reaction of the urea cycle is that 2 molecules of ammonia are combined with carbon dioxide in the form of bicarbonate to form urea and water. You can see ammonia, and carbon dioxide in the middle here, or what used to be carbon dioxide. So in other words, 2NH3 plus CO2 goes to urea, and this notation is just a linearized version of a ketone plus water, or a carbonyl plus water. Urea, on the other hand, can then be safely excreted in the urine. So urea, unlike ammonia, is very non-toxic. I think it has like a LD50 of like 15 grams per kilogram, which literally means that in order for 50% of a population of humans to die from urea poisoning, their bodies would have to be 15 grams per kilogram, which is 1.5% urea. So we're not going to encounter that problem where our body is 1.5% urea by mass. The first step here is going to be formation of carbamoyl phosphate. So glutamate from transamination. Here's our amino acid, which leaves as an alpha keto acid in our alpha keto glutarate, which then becomes aminated to glutamate. Will then enter the liver. So we're in the liver mitochondria now. And then glutamate can then undergo reaction catalyzed by glutamate dehydrogenases, which will oxidize glutamate back actually to alpha ketoglutarate, so they can undergo further reactions. In the process, when glutamate gets oxidized to alpha-ketoglutarate, we lose the ammonium group, and either an NAD or plus or an NADP plus, don't worry about that, it will become NADH or NADPH. We just associate NADP plus and pH with, of course, pentose phosphate pathway. It's a little, maybe send you a little bizarre to see them here, but it's not going to be important for our purposes. Then our ammonium will require hydrolysis of 2 ATP, and we form 2 ADPs with 1 inorganic phosphate in order to ligate a carbon dioxide, remember in the form of bicarbonate, to ammonium. So we're ligating bicarbonate to ammonium to form carbamoyl phosphate, and we actually get to keep one of those phosphates that were hydrolyzed from ATP. So this guy would have extra energy. This is our carbamoyl phosphate molecule here. So if you wanted to memorize an additional fact about the urea cycle, so we said it occurs in the liver, and we said that the urea cycle is for elimination of amino groups from the diet or from metabolism. The other thing you might memorize is that carbamoyl phosphate structure and that it's one of the urea cycle intermediates. I wouldn't say any of the enzymes here are super important. We'll just take a quick peek at the rest of the urea cycle. We kind of covered the major parts. Some other molecules not appearing elsewhere in metabolism that are involved here. Just as an insurance policy, so you don't hit me up saying, hey, you told me not to know ornithine and citrulline, and they were on my MCAT. If you don't necessarily memorize the structures of the names completely, but you just kind of associate, if you see an ornithine or a citrulline, which is some -INE molecule, if you can associate that with amines, then think about the urea cycle. So ornithine here is actually kind of like the 21st amino acid. What amino acid is ornithine most similar to? Which amino acid is ornithine most similar to? So here's our N-terminus, C-terminus, alpha carbon, the 3 CH2 groups, and then an NH3+. Very similar to lysine. So ornithine, while it is an amino acid, there's no codon that encodes for ornithine. So you're never going to get ornithine incorporated into proteins. And this will be the main place where we see ornithine. Ornithine is going to accept, I wish I'd shown the carbamoyl phosphate, but it's going to accept a carbamoyl phosphate to become citrulline, and we'll have a phosphate group leave. And then the rest of these steps are, again, we're not going to memorize in detail what's going on here. Citrulline will combine with it, definitely don't worry about this, combine with aspartate from this other molecule called arginosuccinate. And arginosuccinate will then will split into fumarate, which will then go into the citric acid cycle, and arginine. And arginine, the side chain will then split off. So we'll lose this amino group and this amino group, and or kind of not an amino group yet, but they will become one, as well as the carbon. As well as the carbon is going to become urea. And so if you wanted to memorize a fourth thing about the urea cycle, you could memorize that arginine is the most immediate precursor to urea. Right, so any questions about the urea cycle? Okay. And then our last slide of the day here. And this is just going to be very brief. We're looking at the urea cycle. You can see here's our arginine, here's our urea, here's our ornithine. Ornithine combines a carbamoyl phosphate to form citrulline. Citrulline combines with aspartate to form arginosuccinate. Arginosuccinate splits into fumarate and arginine, and then arginine makes urea. So this is our general process of urea cycle. And then this is how it sort of can connect with the citric acid cycle. Here's our glutamate, which received the amino group of an amino acid, becoming alpha-ketoglutarate, and then aspartate entering into the urea cycle, giving our second equivalent of ammonia, and then our citric acid cycle. And that's it for MCAT Metabolism. Woo, we did it. We made it through 5 lectures together. And does anybody who's present in our Zoom session have any questions while we're still recording for YouTube? All right. Well, for everybody watching this on YouTube, thank you for watching my series of metabolism videos. Feel free to leave a subscribe. Feel free to like some of my videos so that that good old YouTube algorithm will help spread them to more people. And I hope you found them very informational, and feel free to request handouts for any and all of the MCAT Metabolism lectures that we've done. And I will see you on another topic.